Hello, this is Mr. Fahy, and welcome to Mr. Fahy's Science Basement, where today we're going to learn a little bit about genetic engineering. And yes, I have a whiteboard in my basement because that's what it's come down to. Genetic engineering, pretty straightforward to explain, but really hard to do. Genetic engineering is the manipulation of genes in living things to give you traits that you want. Now, we're going to be using a lot of words like genes and DNA and chromosomes, so I thought this would be a great little picture to throw in front of you. Most of you probably remember seeing this picture a few weeks ago, but when we're talking about genes, we're talking about a sequence of A's, G's, C's, and T's that code for one specific thing or a specific protein in a living thing. And those genes reside on these long strands of DNA. In fact, you have feet of DNA in each one of your cells, probably millions of miles of DNA in your entire body. And that DNA coils itself, packs itself up nice and tightly into a chromosome. And in our cells, we have 46 of those jammed up into our nucleus. Different animals and plants and fungi, they all have different numbers of chromosomes, but it's all pretty much the same mechanism. The only one that's a little different are bacteria. Bacteria actually don't have these X-shaped chromosomes. They kind of have these little circles, which we'll be taking a look at the end of this slideshow. And those circle chromosomes are pretty cool because you can do some interesting things with them. When most people think of genetic engineering, they think of something modern. But the truth is, humans have been genetically modifying organisms for a long time. In fact, thousands of years. One of the greatest examples of this is something called selective breeding. If you take a look at the dogs we have today, we have a ton of different variety of dogs, but they all do come from one common ancestor, the wolf. And if we could go back thousands, not even that long ago, thousands of years ago, where wolves, being pack animals, must have hooked up with some folks along the way and started palling around with them. Once those wolves become domesticated, then humans could choose which wolves bred with which wolves. So if you start choosing the shortest of the wolves and always mate the two shortest wolves you've had, eventually those short genes will be propagated and passed down from generation to generation, where you choose the shortest of those and then the shortest of those until you wind up with a Chihuahua. The same thing is true kind of all dog breeds. You can basically pick and choose the parents, and if you keep doing that for generations and generations, you can get a variety of traits that you find favorable. Another example of selective breeding, which is relatively lame, is looking at modern day corn. To tell you the truth, corn like this does not exist in nature and probably never did. What happened a long time ago, probably again thousands of years ago, as folks were trying to eat things as they were walking around, they found this common plant here of the time called teosinte. Now, doesn't have a lot of food to it. It's not really going to sustain you too much. Not too tasty, really doesn't have a whole lot to offer. But as humans started farming, they started to then cross, just like Gregor Mendel crossed pea plants, they started to cross the flowers of these plants selectively. In other words, they would choose the biggest yielding teosinte plant and then cross that with the biggest one. And if you kept going with the biggest one and the biggest one, take the biggest one of those, after generations, thousands of years, in Mexico, we wound up with what is modern day corn. Again, corn like this, not existing in nature without human modification over time. So really, genetic engineering has been going on a while. One of the best examples, I think, would be the dairy cow. This cow's got some issues, but it's got some horns and a big udder and stuff like that. But you get the idea. There is no wild dairy cow. All right, it comes from an ancestral cow type thing, like if you were to take a look at like a yak or a bison, something in that family. But then again, domesticating and selectively breeding over time, you can start to get the characteristics that you want. In other words, these are characteristically easy to find in a field. They also have kind of large udders that produce milk often. Now you have to mess with them to get them to produce milk, but these are genetically engineered animals. They've just been genetically engineered over hundreds, if not thousands of years. Now, this is not what people think of when they think of genetic engineering. They think more about DNA cutting and piecing things together. And that's the piece we're going to look at next. Not this old selective breeding method, but new modern genetic engineering. All right, so here's a more modern form of what you think of when you think of genetic engineering. And it looks really complicated, but to tell you the truth, it's not. And in grad school, I actually did a lab where we got bacteria to incorporate DNA and have different traits than what we started with. So even though this looks really complicated, even your science teacher did this in the later years of college. So it is something that is very doable. So what we're going to do is we're going to look at a genetic disorder called type 1 diabetes, where the human body has a hard time manufacturing insulin. Now, insulin is in a human gene. Okay, the gene for insulin we all have, just some folks have a harder time producing it than others due to a defective gene. So the way that insulin is produced for diabetics is actually pretty cool. It uses some pretty high-tech genetic engineering. Let's take a peek. So what they do is they take the gene for insulin, for making insulin, out of a healthy human cell. So they can actually go into the DNA, use a cool chemical called a restriction enzyme, and snip out or cut only the gene for insulin out. And then isolate it by itself. Now, that gene by itself is not going to do any good. You have to put it into something that can make insulin. And some kids say, well, why don't you take this insulin gene and just put it into somebody who's sick? Well, if you're to put it into someone who's sick, you'd have to put that insulin gene into every single cell of the hundreds of thousands of cells in the pancreas. And that's very, very challenging. So the way that this is done is kind of cool. This down here is a bacteria cell. Now, you might remember early in the presentation, I mentioned that bacteria cells don't have like chromosomes that are shaped like X's. They're actually shaped like circles. And here's a really big chromosome. It's a big, windy, twisty circle. And this thing right here is a super small circle that's in that bacteria. And these little small circles of DNA are very, very, very common. Okay, it's called a plasmid, actually. So what scientists can do is they can actually remove the plasmid from the cell, cut the plasmid open, and insert the human gene for insulin into the bacteria's plasmid. So then the bacteria can be forced to take that chromosome back into itself. Now this bacteria has the gene for insulin. See, you can see it right there. And if you look down here, this is a regular bacteria that doesn't. So now you've genetically engineered a bacteria to produce insulin. So if you take that bacteria, you let it grow, you clone it, you let it grow in culture until you have hundreds of thousands of them, these bacteria will actually produce human insulin for you. And that's how insulin is harvested and given to patients who need insulin. In fact, a huge amount of the drugs that you take for various illnesses are manufactured in this way through genetic engineering. All right, let's take a look at a second and very, very popular form of modern genetic engineering, which is cloning. And we're going to show you a very famous case of cloning in the 1990s of a sheep named Dolly. So this right here is the sheep that we are going to clone. So we're going to make a clone of this sheep right here. Okay. So in other words, we need to take the DNA out of this sheep and produce another sheep that is an identical twin from a DNA standpoint to this original sheep. So check out the way that this was done. It's actually kind of crazy. So what they did is they actually took this sheep and took a cell out of the sheep. They actually took it out of the sheep's udder. Okay. They actually removed the nucleus from the cell of that sheep. And as you may remember from earlier in the presentation, that nucleus holds the DNA. Okay. So now they have the nucleus of the cell, a nucleus out of the cell of a sheep that they want to clone. So then they take another sheep, okay, take an egg cell out of the ovary of this sheep and remove the nucleus. So now we have the nucleus of the sheep we want to clone, and then we have an empty egg cell from a second sheep. Okay. Through a kind of complicated process, the nucleus is inserted into this egg cell here. And then you have a word you might remember, a zygote, a single cell that will eventually give rise to a new sheep. The only interesting part is the cell actually comes from this sheep, but the genetic information actually comes from this sheep in the nucleus. Okay, so that zygote then divides, undergoes mitosis, differentiation, and this embryo is placed into a third sheep into the uterus, and this serves as a surrogate mother for the baby that is not hers, the baby lamb there. And then at the end of gestation, this sheep gives birth to a clone of the original sheep that you had up here. Scientists have had some success cloning other animals, and there's a lot of scientific controversy around it, but there are some possible benefits to being able to do this. One of which, when you think about humans, is not simply making new humans for like a clone army, but you can clone tissue that could be used in the event of injury, heart replacement, and you could actually clone human parts or pieces. So that's something that scientists are thinking about trying to deal with now. But again, it's riddled with controversy, but kind of an interesting thing. So I'll be available to answer questions either through the comment section on the bottom of this post, or if we do a Zoom conference later in the day, you're more than welcome to ask any questions. All right, thanks for coming to Mr. Fahy's Science Basement, where we have a whiteboard.